Welcome to part one of the reproduction outline. The first thing that we're going to look at is a few of the diagnostic characteristics of living mammals. And we've talked a lot throughout the semester about how mammary glands are universal throughout mammals. All infant mammals depend on milk from mammary glands when they're first born as their form of nutrition. So all species have mammary glands. And if we look at mammary glands, the tissue of the mammary gland, the actual histology of the tissue, is very similar from species to species, but there are different ways of structuring the tissue and the collecting ducts and how the milk is delivered to the infant. So in eutherians, if we look at those first, there are two basic types. The first type that we find in a lot of mammals, including humans, has the mammary tissue arranged in little lobules connected to collecting ducts that converge into multiple collecting ducts that empty out through an external protrusion called the nipple. If we look at a contrasting type that we find in a lot of ungulates, like things like cows, they have similar tissue at the cellular level and some collecting ducts, but then those collecting ducts empty into a common space that's called a cistern, and that has a single opening through an external protuberance that's called a teat. We have a few other pictures to show. So here's a picture of a cow udder. So this is the type that has a cistern. And if we look at another diagram that shows the tissues, you'll see the little microscopic structure of the milk-producing cells that are arranged like in little clusters of grapes with collecting ducts that converge and dump into the cistern, which is basically a big holding place for milk. And that milk empties out through the teat. If we look at the primate type of gland, here's a picture of a gorilla. So this is what the primate type gland looks like, and it's also the type of mammary gland that you find across lots and lots of mammals. And the major difference is that there's no big holding area for milk, that the milk is retained within the lobules of the mammary gland until the infant attaches to the nipple, and that triggers smooth muscle contractions that actually squish all the milk out. So if we look at the cells, the secretory cells are actually the ones on the very inside of these lobules, and that's true for the udder type gland as well. There's smooth muscle that overlies the outsides, and when that muscle contracts, the milk that has been collecting inside these structures just gets squished out. It moves out through the ducts, and then there are multiple ducts that open out through the nipple, and the milk is delivered to the infant. If you're interested in histology, maybe pathology, you want to have a career that deals with things at a microscopic level, you might be interested in looking at these slides of mammary tissue. Whoops, wrong direction. Let's get bigger. So this is inactive mammary gland tissue, and you can see the secretory cells here. They kind of look like clusters of grapes, and since this is a two-dimensional slice, what you see is circles of various shapes, but they are three-dimensional, kind of bubble-shaped structures, and milk would be produced by the cells in this structure and collect on the insides, but this is the inactive gland, and so there's no milk. What you see mostly is a little bit of secretory tissue, but a lot of connective tissue, so that's all this pinkish stuff in between. So the inactive gland is not producing milk. It's got a bunch of cells that could produce milk, but most of the structure is taken up by connective tissue. If we look at the active tissue, we see something very different. and that is that the connective tissue has now been compressed, so it's much less visible. So it's still the same pink stuff here, but it's just squished in between all the different lobules that contain the secretory tissue. And the secretory cells here are shown as these circles, and they're producing milk, and the milk here is stained pink. So all of this is milk that's collecting inside of these alveoli. One of the interesting things about this picture is that it shows you how some sections of the gland may be completely emptied of milk and other sections of the gland may be full of milk. So depending on the section, when the infant starts suckling, the milk will squish out of a set of these alveoli, and they'll become empty, and then over time they'll refill with milk. So this is the active gland again. You can see how it's completely composed of the secretory cells and spaces for the milk to accumulate, and the connective tissue is relatively small. Alrighty, the second reproductive structure that is diagnostic of mammals is the complex placenta. Monotremes don't have a placenta because they lay eggs. Marsupials have a related structure. It's a placenta, but it's not super complex, and we'll kind of go over the differences between the two in a little bit. The complex placenta has several layers of tissue. This is an armadillo placenta. It's a little weird in that it has a single outer membrane and it contains 4 fetuses. And these 4 fetuses are identical quadruplets. So you can see they have their own inner membrane that's called the amnion, but their other membranes are shared. And for this reason, this is just a bit of trivia. Armadillos are sometimes used in biomedical experiments because you have sort of a control group. You have 4 genetically identical individuals that you can follow and learn from. The other example of a placenta I wanted to show you is a human placenta, because you don't get to see that very often. It probably doesn't look like you expect it to look. So this does not show the part where the fetus is located. It just shows the part that attaches to the inside of the mother's uterus, and that's this disc right here. And you can see that it's very vascularized. So this is the umbilical cord. It would be attached to the developing fetus, and blood flows from the fetus to this part of the placenta and then back again. This section is where all the gas exchange happens and where all the nutrient exchange and so on. All right, in the next section of the lecture, we're going to compare the different groups of mammals, the monotremes, the marsupials, and the placental mammals, and look at how they reproduce and some of the major differences between them. Obviously, there are some similarities between the groups. All of them have internal fertilization. All of them nourish their infants with milk from mammary glands, but they differ pretty substantially in how they reproduce, what the infants are like when they're born, how quickly they develop, and so on. So we'll start first with the monotremes and talk about how they are born and developed, and then we'll talk about marsupials and placentals. So monotremes, as you know, they lay eggs. The eggs are either deposited in a nest, like birds, or they are carried against the mother's abdomen in a groove in the abdomen that is sort of pouch-like. They're not completely enclosed, but they're kind of stuck there. There are some sticky secretions that make sure the eggs stay there in the groove. Another interesting fact about monotremes is that they have no nipples. So when offspring nurse, the milk is just it just oozes onto the skin or onto the hair, and then the infants lick it up from there. They have no placenta, And the eggs have actually a very short time to hatching. So what that means is that the young are born highly altricial. Altricial means helpless and very undeveloped. I'll show you some pictures and that will make it clear what I mean. So if we look first at the platypus, one example of a monotreme, they lay one egg at a time, usually maybe two eggs. And the egg is very, very tiny. So here's somebody holding an egg in the palm of their hand, and that's in comparison with a penny, so smaller than a penny. It takes 9 days of incubation before that egg hatches, and here's what it looks like in hatching. So here's the egg, and here's the infant echidna. You can see its little feet and its little head, but it's very undeveloped. So you can see right through the skin, you can see blood vessels, you can see the eyes are just black dots. You can't see any of the eye structures there. Obviously it can't locomote, it can't get around. So it's very, very undeveloped. But it develops fairly rapidly over time. So right after hatching, it's going to live in its mother's pouch for about 12 weeks. It's going to get spines. There's their baby echidna. You can see the pouch or the groove in the mother's abdomen there. They develop spines. Sorry about this. It keeps bumping me back to weird places. Spines at about 8 weeks, and at about 12 weeks, the infant's eyes open, and at that point, the infant leaves the pouch and stays in a burrow, and it's not weaned for 20 weeks. So some quick pictures. Here's the baby echidna just getting its spines. And then here's what they look like when their eyes first open. Alrighty, we'll also look at the platypus, the same basic thing, 1 or 2 eggs that are very, very small, and the infants when they hatch are very undeveloped. And they also grow very slowly, so I'll let you click on the links and see the pictures on your own time, but they first get hair at about 7 weeks, their eyes open at about 9 weeks, and they're weaned at about 16 weeks. So the bottom line here, I don't care that you remember how many days it takes for the eggs to hatch or how many days it takes for them to be weaned. They have a fairly short incubation time, only a few days, but they have a very, very long time after they're born where they're dependent on maternal milk. All right, let's look at marsupials now. So they give birth to live young, so no more eggs, but the young are born very, very undeveloped. So they're also highly altricial. Once they're born, they attach to nipples, either in the female's pouch, if it's a species that has a pouch, or just hanging from her abdomen. About half of marsupial species don't have a pouch, so the infants are just carried on the outside of the mother's body. The placenta is fairly simple, so it consists of a few layers of membranes, which we'll take a look at in a minute, but it's not as complex as what we see in eutherian mammals. That's why we call them placental mammals, because they have the highest form of development of the placenta. But there is variation in different species of marsupials, so some marsupials have a more complex placenta than others. They are all born highly altricial, which means that they're very small relative to the size of the adults. So if we look at a marsupial that's sort of cat size, the adult is about 4,000 times bigger than the infant. If you look at a kangaroo, the adult is about 33,000 times bigger than the infant. So the infant is very, very small, like the size of a bean or a grain of rice in comparison to the adult. And the size and the lack of development comes down to a very short gestation period. So they're only retained in the mother's uterus for a few days to no more than 6 weeks. This lack of development means that much of the physiological structures of the infant are incomplete. So the heart is not four-chambered yet. And the lungs don't have the structures, the bubbly structures that gas exchange occurs across. I'm going to pause here and pick it up in the next section so that they're all short videos. So I'll be back with you in a minute.